Okay, hello to all of you. Let's start discussing chapter number 20th. And in this chapter number 20th, we are going to discuss how the high energy electrons which are there in the food, they go through different proteins which are present in the inner membrane of mitochondria, and they flow towards the oxygen. And on the way, when these electrons, these high energy electrons, so till now we have covered up to Krebs cycle, and we have seen that how the high energy electrons which are present in the food, they are carried by electron carriers like NAD plus and FAD, and they get reduced to NADH and FADH2, and they bring these electrons to the inner membrane of mitochondria, because in the inner membrane of mitochondria are embedded some of the very important protein complexes, and these protein complexes form the electron transport chain, and at the end of the electron transport chain there is oxygen, and oxygen is very electronegative and it pulls the electrons towards itself. And on the way, when these electrons flow through these proteins, and these are special proteins, we are going to discuss these proteins also, and on the way some of the protons are pumped towards the intramembrane space, and which results in the higher concentration of the protons in the intramembrane space of the mitochondria, and then this results in the formation of proton gradient. So there are more protons concentrated in the intramembrane space, which is the space between outer membrane of mitochondria and the inner membrane of mitochondria, and then these electrons, sorry, not the electrons, the protons, they fall back through another protein, which is an enzyme called ATP synthase, and that results in the formation of a lot of molecules of ATP. So this process by which these protons, they fall back through ATP synthase, this process is called chemiosmosis. And electron transport chain, and this whole phenomena of the flowing of the high energy electrons through the proteins, which results in the pumping of protons in the intramembrane space, and then the flow of the flow of the protons down the concentration gradient, from the higher concentration to the lower concentration, it results in the activation of this enzyme, and that results in the formation of a lot of molecules of ATP. This whole phenomena is called oxidative phosphorylation. So in this whole process of aerobic respiration or cellular respiration, ATP is produced by two different phenomena. One is called substrate level phosphorylation, and second is called oxidative phosphorylation. So the molecules of ATP which are produced in glycolysis as well as in the Krebs cycle, they are produced by the process of substrate level phosphorylation. So I would like to repeat here one more time. Substrate level phosphorylation is a process in which ATP is formed from ADP. So adenosine triphosphate is formed from adenosine diphosphate. The donor of phosphate is some intermediate metabolite, and this process takes place in the presence of some enzyme. Like last step of glycolysis, if you remember, phosphoenol pyruvate is converted into pyruvate. So in this step, ADP, adenosine diphosphate, is converted into adenosine triphosphate. So ADP is getting converted into ATP, and the source of phosphate is phosphoenol pyruvate. So what is phosphoenol pyruvate? It's an intermediate metabolite, and this reaction takes place in the presence, or this reaction is catalyzed by the enzyme called pyruvate kinase. So this is substrate level phosphorylation. So in this chapter we are going to talk about the process called oxidative phosphorylation. So this is a whole phenomena by which a lot of molecules of ATP are produced in the cellular respiration. It involves electron transport chain as well as it involves chemiosmosis. So that's what we are going to focus in this chapter. So this is all what I just explained. So I would like to explain you one more time, looking at the structure of mitochondria. So this is outer membrane of mitochondria, and this is the inner membrane of mitochondria. So these are called, these invaginations, they are called cristae. Okay, and the protein complexes, the four protein complexes which we are going to discuss very shortly, And the important thing to know about these protein complexes is that these protein complexes, they have proteins in them, and these are special proteins because they have some metal ions in them. And what's the significance of these metal ions in these proteins? The significance is that these proteins, because of these metallic ions, they can accept electrons as well as they can lose electrons. Because of the presence of these metallic ions in them, you will see that they have copper in them, they have iron in them. Because of that, and we will talk about some special proteins called cytochromes, they have iron in them in the form of a group called heme group. So because of these metallic ions in them, they can either accept electrons or they can lose electrons. So they can get reduced, they can get oxidized. And because of these metallic ions, these proteins, these metallic ions can get reduced and oxidized. And because of that, is possible, because of this property of these proteins, the flow of electrons through these proteins is possible. And remember that at the end of the electron transport chain there is oxygen, because this oxygen is the final electron acceptor. So these electrons, you will see that. So I will show you, I will just very shortly see. So this is the inner membrane. So outer membrane, inner membrane, this area is called mitochondrial matrix. This region is called intramembrane space or intermembrane region. And the protein complexes are embedded here in the inner membrane of mitochondria, and these electrons will flow through the proteins which will be embedded here. And as the electrons will flow through these proteins, these electrons are high energy electrons. So the energy which is associated with these electrons is actually used to pump the protons from this region to this region. So here is low concentration of the protons. Here is a high concentration of protons. So when these protons, they have to be moved from this region where there is a low concentration, and this region is a region with high concentration of protons. Energy is required for this process because this is called active transport. So let me explain you quickly what is active transport and what is passive transport or diffusion. So diffusion or passive transport is when the molecules or the ions, they fall from high concentration to low concentration. So when the molecules or the ions, they fall from high concentration to low concentration, this is a natural process. This is diffusion. When the molecules are moving from high concentration to low concentration, this process is a spontaneous process. This process does not need any energy. But when the molecules or the ions, they have to move from low concentration to higher concentration, they need energy. So that's an active transport. So that is the same thing which is happening here, that this region has low concentration of protons, and this region has high concentration of protons. This region has low concentration of protons, this region has high concentration of protons. So when these electrons, these high energy electrons, they flow through these proteins, when these high energy electrons, they flow through these proteins, the energy which is associated with these electrons is used to pump these protons from this region to this region. So as a result of that, there is, so on this side, this is intramembrane space, this is matrix. So there is a collection or the accumulation of protons in this region. So more protons here, less protons here. So that results in the formation of a proton gradient. That results in the formation of proton gradient. And so the energy which is actually in the form of high energy electrons, and these high energy electrons which are coming from the food we have eaten, that is used to make the proton gradient. Okay, and then there is another enzyme involved in which is embedded in the inner membrane of mitochondria, which is called ATP synthase, and these protons, then they can diffuse, okay, because there's a high concentration here, low concentration here, they can diffuse through that enzyme, and that process is called chemiosmosis. That will activate ATP synthase, and that will result in the formation of a lot of molecules of ATP. So so I think I have already explained all these things. Okay, this basically shows you the flow of electrons through electron transport chain. So we know that we have in our cells we have two different kinds of electron carriers. One is NAD plus, which gets reduced to NADH. Then we have FAD, which gets reduced to FADH2. So these are some of the proteins. I will be showing you exactly what protein complexes are. So so NAD plus basically delivers its electrons to the protein complex number 1, and then this protein complex number 1 delivers these electrons to coenzyme Q, and here FADH2 also delivers its electrons to coenzyme Q, and then there are various cytochromes involved in this whole electron transport chain. We are going to discuss all of them in detail, and these electrons, they flow through these protein complexes, and then eventually these electrons they reach oxygen. So when they reach oxygen, these electrons, they are low energy electrons because the energy is already used to create the proton gradient, and oxygen, when it accepts these electrons, these low energy electrons, oxygen gets reduced and oxygen forms water. So carbon dioxide and water, these are two byproducts of this process of cellular respiration. So water is, this oxygen gets reduced, oxygen accepts these electrons, oxygen accepts these electrons and it gets reduced to water, and carbon dioxide is released in the various steps, and this is the carbon dioxide which we exhale. So this is basically showing you all the protein complexes. This is complex number 1, complex number 2, complex number 3, complex number 4. So in the complex number 1, there are some proteins which have, there are some proteins which are called flavoproteins, and they have FMN as the coenzyme, flavin mononucleotide, and there are some proteins which have iron and sulfur in them. We call them iron and sulfur proteins. So they're part of complex number 1. Then this is your complex number 2, which has iron and sulfur proteins in them. And then this is complex number 3, which has iron and sulfur. Complex number 3 have proteins which have iron and sulfur in them. Also in the complex number 3 there is cytochrome C1, and this is complex number 4. Complex number 4 has cytochrome A, cytochrome A3 and copper in them. And this is basically showing you the flow of the electrons, how the electrons they are delivered. So this is complex number 1, complex number 2, complex number 3, complex number 4. So complex number 1 is called NADH coenzyme Q oxidoreductase. So NADH is an electron carrier which is bringing the electrons from glycolysis, from the conversion step, as well as Krebs cycle, and it delivers the electrons. So NAD plus is in the form of NADH. It delivers the electrons to complex number 1, and NADH gets oxidized to NAD plus. Okay, and these electrons, they go through flavoproteins. It goes through iron sulfur iron sulfur containing proteins, and NADH, when it delivers its electrons to flavoproteins, NADH itself gets oxidized, it reduces these proteins, and then this protein delivers the electrons to iron sulfur proteins. Then at that point, these flavoproteins, they themselves gets oxidized, and they reduce these proteins, and then this complex number 1 delivers the electrons to coenzyme Q. Okay, and then this coenzyme Q also gets the electrons from complex number 2. Complex number 2 is called succinate coenzyme Q oxidoreductase. So complex number 2, it actually passes the electrons from succinate to coenzyme Q. So both these complexes, they are delivering the electrons to coenzyme Q. And same thing is happening in complex number 1 also, that FAD, which is the electron carrier, it brings the electrons, it carries the electrons, and it is itself reduced to FADH2. And this FADH2, which is reduced form of FAD, it delivers the electrons to the proteins which are present in the complex number 2, and these proteins, they have iron and sulfur in them. So when FADH2 itself gets oxidized, it reduces the proteins which have iron and sulfur in them, and then these iron and sulfur containing proteins, they get reduced, then they transfer the electrons to coenzyme Q, then coenzyme Q gets reduced, and these proteins, they get oxidized. So in this whole electron transport chain, one protein or one electron carrier is delivering the electron from, in this whole electron transport chain, this is what is happening. So one protein or electron carrier which is reduced, it delivers the electrons to the next one, and it itself gets oxidized, and it reduces the next one. And eventually in this whole electron transport chain, the final electron acceptor is oxygen, and oxygen actually finally accepts the low energy electrons, because in this whole process, the high energy electrons, they deliver the energy, and that energy is used to pump the protons from the mitochondrial matrix to the intermembrane space of mitochondria, and that results in the formation of proton gradient, and that proton gradient is responsible for the activation of another enzyme called ATP synthase, and that results in the formation of a lot of molecules of ATP by the process of oxidative phosphorylation. Okay, coming back here, complex number 3 is called coenzyme Q. So you can see it's written here, coenzyme Q with 2 hydrogens, which means it is a reduced form. So coenzyme Q received the electrons from this complex, it received the electrons from this complex, and it got reduced. So the reduced coenzyme Q delivers the electrons to cytochrome C. So here is cytochrome C. Okay, and complex number 4 is called cytochrome oxidase, because it passes the electrons from cytochrome C to oxygen. And in this complex, there are 2 cytochromes, cytochrome A and cytochrome A3, as well as there are proteins with copper in them. Okay, so all these cytochromes, they have iron in them, and that iron is in the form of heme group. So that is what is overall happening in this electron transport chain. Okay, now we are going to talk a little bit in more detail about these complexes. So complex number 1, as I just said, it is called NADH coenzyme Q oxidoreductase. It gets NADH to coenzyme Q. This protein is an integral part of the inner membrane of mitochondria. It is embedded in the inner membrane of mitochondria. It includes several proteins that contain iron sulfur cluster and the flavoproteins, and those flavoproteins, they have FMN as the coenzyme, which is called flavin mononucleotide, and here is the structure of FMN. So this basically shows you how the electrons, they are getting transferred. So this is NADH, which is reduced form. It is going to deliver the electrons to the flavoprotein, which has FMN in it. NADH, it gets oxidized and it gets converted into NAD plus, and it reduces flavoprotein. And then this reduced FMN, it is going to reduce the protein with iron and sulfur in it, and it is itself going to get oxidized. So this is all, through this all electron transport chain, it's all oxidation reduction reactions taking place. And the reduced iron sulfur, it's going to donate its electrons to coenzyme Q, and this coenzyme Q is going to be reduced to coenzyme Q H2. This is the reduced form of coenzyme Q, and this is your coenzyme Q. You can see here how it gets reduced. So this is the coenzyme Q. You can see here it's oxygen group here, oxygen group here. When it gets reduced, it gets H here, it gets H here, and this is the reduced form of the coenzyme Q, or it's also called reduced hydroquinone. Coenzyme Q is also called quinone, and the reduced form is called hydroquinone. Okay, this is same thing. It's a similar pattern. I'm going to be going a little fast now because the same thing happening in all through all the complexes, but all of them have different components in them. So this is complex number 2, and complex number 2 is called succinate coenzyme Q oxidoreductase. It catalyzes the transfer of electrons from succinate to coenzyme Q. So succinate is oxidized to fumarate, and this is a reaction which we just studied in in previous chapter, in chapter number 19. This is a reaction in Krebs cycle. So succinate gets it's oxidized to fumarate, and it takes place with the help of the enzyme called succinate dehydrogenase. And this is the FAD is the basically the coenzyme in the succinate dehydrogenase, and this basically carries the electrons and it gets converted into FADH2, and then this flavin group is reoxidized, and it delivers the electrons to the protein with iron and sulfur in it. And then these proteins with iron and sulfur in it, they get reduced, and then they pass the electrons to coenzyme Q. And then in the complex number 3 and complex number 4 are involved some proteins. These are special proteins which have iron in them. So they're called cytochromes. So these have iron in them in the form of a group called heme group. Okay, so cytochromes, they are groups of heme containing proteins, and they have iron in them in this form, in these two ionic forms. So it can be ferric ion. So ferric ion means Fe+3, and when it accepts electrons, it gets converted into ferrous ions. So ferric ion gets reduced to ferrous ion, and when this ferrous ion, it loses electrons. So here ferric ion is gaining electrons, so it is getting reduced to ferrous ions. And when ferrous ions, they lose electrons, they get converted into ferric ions. So in that situation, they are getting oxidized. And there are different kinds of cytochromes, A, B, and C. We will be coming across them. So in the complex number 3, the electrons from the reduced coenzyme Q, they are through this complex. This complex is called reduced coenzyme Q or hydroquinone cytochrome C oxidoreductase, because the electrons, they are passed through the reduced coenzyme Q to cytochrome C. And in this complex, there are iron sulfur proteins, as well as there is a cytochrome C1. So this is complex number 3, you can see here. Okay, so in the complex number 3, there are these cytochromes. There is a cytochrome C1, there are iron sulfur proteins, and also there is cytochrome B, and this cytochrome B can be cytochrome Bl or cytochrome Bh, and the electrons, cytochromes, can be passed in a cyclic way, and this is called Q cycle. So coenzyme Q, as you saw that when it is reduced in that form, it has 2 electrons with it, that is shown here. It accepts 2 electrons. So in the reduced form, the coenzyme Q has 2 electrons in it, and it can pass 1 electron through these proteins, and these proteins then can pass the electron to cytochrome C1, and then from cytochrome C1 it can pass the electron to cytochrome C, and the another electron can go through these chromes in a cyclic way, and this is called Q cycle. And in this whole process, when these electrons are being passed, remember that when all these electrons are being passed through these protein complexes, some of the protons are from mitochondrial matrix to intramembrane space. So this is intramembrane space, this is mitochondrial matrix. Let me go to this slide and show you exactly what is happening when these electrons are getting transported. So this picture actually shows you that when these electrons, they are getting transported through these protein complexes, there are these protons being pumped. So through complex number 1, 4 protons are pumped. Through complex number 3 also 4 protons are pumped, and through complex number 4 2 protons are pumped. So when the electrons are being passed through these electron carriers in the electron transport chain, around 10 protons. So when the 2 electrons, they are delivered by reduced NAD plus, which is NADH. When 2 electrons, they are delivered to the first complex, and these electrons flow through all these protein complexes. In total 10 protons are pumped in the intermembrane space. So coming back to complex number 4. So complex number 3 I have already explained, and it is all same things written here what I just explained. Complex number 4 is called cytochrome C oxidase. Why? Because cytochrome 4 transfers the electrons from cytochrome C to oxygen. That's why it's called cytochrome C oxidase. So it transfers the electrons from cytochrome C to oxygen. So this is complex number 4, you can see. So this is cytochrome C. So complex number 3 passes electrons to cytochrome C, and then this cytochrome C passes the electrons to complex number 4, and in the complex number 4 there is a cytochrome A3, and also complex number 4 have some proteins in which they have some copper in them, and eventually through complex number 4 the electrons are going to be accepted by oxygen, and oxygen is going to be reduced to water. So this is what is happening through the electron transport chain, and this is what I have just said. You can go over this. It's the same thing what's written here. So now, as I said, all these electrons which are flowing through these proteins, there is a pumping of protons also. This is also happening, and it is resulting in the formation of proton gradient. And these protons, they are eventually going to flow through, they're going to diffuse through this protein called ATP synthase. And this ATP synthase is an enzyme, and it has 2 parts. One part is called FO, another part is called F1. Now FO is the part of this protein which is embedded in the inner membrane of mitochondria. So this part is called FO, and this part is protruding out, okay, and this is called F1, and this part of the protein is protruding out in the mitochondrial matrix. So FO, which is embedded, so I'm talking about this, which is embedded in the inner membrane of mitochondria. It is made of 3 different kinds of polypeptide chains, A, B and C. And F1, which is protruding out, which is projecting out in the matrix, it's made of 5 different kinds of polypeptide chains, which includes alpha. There are 3 alpha chains, 3 beta chains, 1 gamma, 1 this is called sigma, sorry, it's called delta, and this is called epsilon. So basically this, if you understand that this is the inner membrane of mitochondria, this is the intramembrane region, this side is the matrix. So F1 is on this side. So in the intermembrane space, which is this, there is a lot of protons here, okay, and there are less protons here, more protons here, less protons here. That results in the formation of proton gradients. Now the protons are going to flow from FO, and they are going to enter into FO, and then they are going to move through this F1. And when that happens, when these protons, they flow back, when they pass from FO and they go through F1, on this process they actually change the conformation of the F1, okay. So this F1 part is made of 3 alpha chains and 3 beta chains, and these the beta subunits, they actually go through conformational change. I'm going to show you, there are 3 different conformations of the beta subunit. So they go through different conformational changes, and that results in the formation of ATP. So the beta subunit, so this is all what I just said. So the beta subunit, so this is what I just said actually, coming back to the same thing which I showed you right now. It's in a different way. So this is the outer membrane of the mitochondria, this is the inner membrane of mitochondria, this is the intermembrane space. Here is accumulation of lot of protons, and this is ATP synthase, this is FO, this is the part of the protein which is embedded in the inner membrane of mitochondria, this is the part which is protruding out. So these protons, they can pass through this ATP synthase, they pass through this ATP synthase through the F1, okay, and they go through FO and F1, and they activate F1, and that results in the formation of ATP. So this process of the passing of protons, the process of passing of the protons from intermembrane space to the mitochondria matrix through ATP synthase is called chemiosmosis. And this, so the F1 part of the ATP synthase has 3 beta subunits, and these beta subunits can exist in 3 different conformations. One is called O, which is open, another is called L, which is called loose binding, and third one is T, which is tight binding. So you can see that these beta subunits, they can exist in 3 conformations. So this is L, this is light binding, sorry, a loose binding, not light binding, it's called loose binding. So this is a loose binding site for the binding of ADP and the phosphate, and then this is the T binding. Okay, so basically O conformation is has a when the subunit, when the beta subunit, there are 3 beta subunits, when it is in the O conformation, it has low affinity for the substrate, and when it is in the L conformation, in this conformation it is not catalytically active, and in this conformation ADP and phosphate binds to the beta subunit. And the last one is T binding, or it's called T conformation. In this conformation, the beta subunit is catalytically active, and ATP is formed is actually bound in this conformation. So you can see the 3 conformations. This is L, loose, T, tight, and this is O. Okay, so you can see here this is binding. So in the loose binding, and it is loose. It's a binding, loose affinity binding conformation, and in this conformation, ADP and phosphate binds here, and this is the T binding, which is the tight binding, and here ATP is bound in this conformation. So you can see binding here in the T conformation, ATP is bound here, in the T conformation it is bound here, and now in the next you can see that the T conformation is changed into O conformation. So you can see the shapes. So this is the shape of the O conformation, and in this you can see the T conformation, the T beta subunit, its conformation is changed, and its conformation is now changed into O, and O is the low affinity binding. So which means that in the O it cannot keep anything bound with it. So when it converts into O site, O conformation, it releases ATP. Okay, and you can see here the L site, because in the L site, in the not the L site, it's L conformation. In the L conformation, ADP and phosphate binds. Okay, and that is now changed into the T site. From ADP and phosphate, ATP is formed, and the conformation of L site is now changed into T conformation, and then after this the conformation of T will change into O, and then ATP will be released. So basically this shows you the overall process, that how ATP is produced by the process of oxidative phosphorylation. And in total, if we start from one molecule of glucose, in total 30 or 32 molecules of ATP are produced. So why it's called 30 or 32? Because different books they give different values. So basically, in glycolysis, 2 molecules of ATP are produced. In Krebs cycle also, it operates 2 times, it goes over 2 times, 2 molecules of ATP are produced, and around 28 molecules are produced in the last stage of electron transport chain and chemiosmosis, and 28 molecules of ATP are produced by the process of oxidative phosphorylation. And 4 molecules, 2 from glycolysis, 2 from Krebs cycle, they are produced by the process of substrate level phosphorylation. So this is all about the last stage of cellular respiration. If you have a question, please let me know.